All right. Hi, everybody. So today's is skeletal muscle. So it's a bit of a follow-up on EMG. We're just going to actually see a couple elements that can change muscle response. So we'll have three main experiments. One is recruitment. Two is frequency, like how quickly stimulations can come in. And then the third one is going to be length tension. All of these are really important because we spend a lot of time in this lab and in this class on the heart. The heart being a muscle that is going to be related to all these concepts, even if it's going to have a bit of a different response in some cases. So learning it in the typical skeletal muscle way is going to help a ton. Okay, so we've covered a lot and seen all this and how each of this, how each of these little things works. And when you zoom in, you can see like just how tiny these little things are, little myofibrils, and how they like bunch up in a muscle, and how when you recruit more of these, you're going to have a bigger response. So we saw that ultimately in the EMG lab, where even though we were mainly starting with that initial stuff, like the actual physiological process of firing one of these and making sure, for example, that this is going to go in and contract in just like that in the sarcomere. Go here. Even though we were looking at this process small scale, which is good to start. Today, we're kind of going to look at the macro of this and how in a large muscle, which is our frog leg, how are all of these cells going to combine, or all these muscles, and going to combine and show us what we need to see in terms of frequency and length tension. So everything up to this point in this section has kind of been done with EMG, right? So we're pretty good here. Hopefully, let me know if there's a couple things that aren't doing so hot. We'll go from there. A lot of the details that we'll get to see today, in a lot of cases, go here at the binding. So when we look at length tension or we look at frequency and why muscle recruitment and response has such a different reaction than just straight up neurons, a lot of those details are going to be found here. So long as you feel pretty good about how this basic idea is working, you'll be fine in lab. The main questions that you'll see on the exam for lab would be much more about the data and the experiments that we're about to see in a sec. So a couple examples, and again, these could technically have belonged in the EMG. I can't remember if this is in the EMG video, so skip over the slide if it is, but if not, it's a really good example because this is typically how you can assess somebody on something. So if we're looking at something like botulism, what's going on here is that typically you have acetylcholine that's going to be shared out from the neurotransmitter. It's going to bind nicotinic receptors, those who are going to allow sodium to come in. And now we've got our action potential of the muscle. Now, what botulism does, botulism toxin, is it kind of makes a little border right here. All this acetylcholine that's in here is no longer invited to come out. No acetylcholine, no response, no contraction. Now, as a disease, this is bad because obviously it gets to the wrong places. Your muscles stop contracting very, very well. As a commercial product, it prevents chronic wrinkling, things like that. So a really good example, though, is going to be the tetanus toxin. And now, if we're talking about disease, this is a super deadly disease. Otherwise known as lockjaw, this is when contractions don't stop. This is when a contraction is also involuntary, when it's supposed to be a voluntary thing. So in our normal mechanism, we actually have something called GABA and glycine to a degree that actually stops the muscle reactions that we've been studying. Because eventually, right, if you're like flexing a muscle, you have to unflex, you have to relax, right? This is typically told by another motor neuron to play stop. What tetanus does is block this interaction. Thus, contraction has a very difficult time stopping. And sometimes contractions will start initiating without, voluntary response, things like that. This is why, yeah, if you step on a nail, go ahead and get like a tetanus booster or at least check. That's basically the idea. Okay, so on to the main stuff. One of the interesting things is, and we covered this in EMG, but it's that if you are lifting something small, so like 3, for example, you have motor neurons that are going to come in and say like, okay, like we're around this whole muscle fiber. Like remember, you have a motor neuron up here that's basically saying like, okay, like we can like innervate this muscle. We can basically give acetylcholine and say activate. But how many pieces of that muscle do you want activating? If you're picking up something small, you only maybe want this, 2%, 5% of what your muscles could do in that case. Pick up something small. You don't need to, fling something small like it with a massive response. Now, in the other case, your motor neurons, which are all around these muscle fibers, they can instead send the signal to say, okay, we need to fire everything we can right now. Activate 100% or close to 100% of the muscles in your area, in this area. This is going to be a response if you're lifting something heavier, in that case, in a lot of cases. Okay. The similarities, and we saw this in that title screen, like to keep track of similarities between neurons, muscle cells. It's a good way to study, compare and contrast. It's also a really good way to assess on exams. So just kind of giving you that. We have the same idea with a threshold, which is there's certain stimuli that for muscles, it's just not going to activate any of the cells as far as an active potential, like something so light that, at least doesn't even do anything. There has to be at least a little bit of resistance to cause some recruitment. And that's the idea behind this word, is that you have a big muscle to recruit from. You have, let's say, 100 little fibers. Obviously, it's more than that. But you may only need a few of those fibers to get something done. If you want to get to the max, though, you will be recruiting 100% of your available muscle fibers if the stimulus is strong enough. So if you want to lift like a desk or something, it's going to say like, okay, like let's get this whole thing up. In this case, this stimuli right here, similar to the neurons, and because I should show you right here, we're stimulating the nerve down here. So we have our threshold right here. It's met right here. That matches. Similar to the neurons, even though we've increased the stimuli for these ones and these ones, it doesn't matter because you're already recruiting 100% right here in all of these. So even if you try, or if we stimulate our frog leg to go beyond 100%, you can't go beyond 100% of what your muscles are made of already. They're all already firing. So in this case, again, that maximum stimuli is going to be the one that initially gets 100%. So similar to the neurons, once you start leveling off right here, it's time to go find and say like, okay, here's my max voltage. Because remember, it's not these two because they were over max. So if you see something on the exam where it's like, what's the response here? It's the max response. This is still the max response. It's just got way more stimulation coming at it. It's still only going to do the same amount of like push that a muscle can do. Okay. But cool thing with these is, again, you get to decide how much of your fibers you're going to like kind of pulse and activate in this case. So in that case, I have a little break right there. Main thing, yeah, I mean, I was going to use this space for something else, but I want to move on. Okay. Experiment 2 is going to be frequency now. So everything we covered before in experiment 1 is all talking about how much of a muscle are you going to be using. And the greater stimulus you need, the more. Experiment 2 is going to be a little different. We're going to keep the amount that we're pushing on the frog muscle the same. But we're going to say you're going to fire at specific intervals. Specifically, what we're going to control is this period of time between each stimulation, right? See how this spread right here, it's pretty far. So what happens with muscles is that if you fire them and you allow them to reset and fire them again, everything's the same. Everything's fine. So let's assume that this response that we're seeing up here, so like this amplitude right here, this is basically your response, which is a good measure of how much you recruited, basically. So we'll actually keep this kind of in the middle on the frog leg. So twitches is what we call when there's plenty of time to reset, everything's good, and you just do the motion again. In exercise, this is kind of just like a slow, normal rep, let's say. Now, if we increase frequency, something strange happens. And this is going to be a big difference between frequency of action potentials in neurons. They don't do this. They don't build. They don't get bigger and more strong, basically. Muscles do. So as we increase, so in this case, the spread between these two is now getting smaller than what we had up here. This spread was still pretty big. So what we're doing here is we've moved on to a second stage. This is something called trepe. In this case, we have not left enough time for the muscle to fully reset. And what I mean by reset is this giant cycle that we've talked about before. All the neurotransmitter action potentials, everything that we talked about, this process hasn't quite ended if you don't give your muscles enough rest. Now, that's not necessarily a bad thing. What this means is that if you increase the frequency of stimulation and like you do like a faster exercise, for example, your muscles actually kind of build up to a higher strength and tension in that case. They're going to be doing more because you're doing something quicker. So in this case, each time we keep moving up, up, and up. Specifically, why is that happening? Why, when we increase the speed of something, is that going to happen? And it goes all the way back to kind of the system, the chemistry. Remember the role of calcium coming out of that sarcoplasmic reticulum. That's triggered by the action potential that hits the muscle cells. Right here, let's look at this trepe right here. So right here, you've stimulated a lot of calcium to come out. By the time you're here, you kind of reset, but some of that calcium is still around. You basically, oh my God, sorry. You basically have some leftover calcium that's outside the SR. It's like kind of still ready to fire again. So if you have like a little extra calcium, the next time you hit that stimulation, we're going to get a little higher, right? Let's say I have this much calcium left over now. Stimulate, get a little higher. We still have this much calcium left. Stimulate, get a little higher. Eventually, like anything, you level off to your max possible. You've recruited 100%. So that's kind of the cool thing with calcium is that, and it's a unique thing with neurons that you don't deal with neurons at all. This is a muscle thing. Is that in this system, when you have calcium coming out of the SR, sometimes it stays out if you stimulate the muscle quick enough again. That's the kind of the cool thing here. The other interesting thing is that as muscles do work, they get hotter. When it's hotter, in any chemical system, things move faster. More calcium is going to show up and, the chemistry is going to move a little faster. That's why we keep ascending up. Now, the key difference with trepe here, yeah, we're making an ascension. We are technically resetting at least a little bit, though. So let's up the ante. Two more options here. Summation is kind of like what we just saw with trepe, but you don't get to reset. So right at this stage, if we stimulate one more time, so much calcium is still left over that we may as well go a little higher. Same thing in each case, right? So long as you quickly stimulate something and keep that muscle still keep all that calcium outside of the SR, it's ready to use it at any time. So it's going to build a bigger response when you give it an equal stimulation to go again. So this, as far as exercise go, this is kind of like pulsing. So this, I'll show you the example in class, but it's almost like if you're doing a lift, but you're or some sort of running or exercise or movement or anything, and you're not resetting pretty much at all, but you're still kind of moving with it. That's how you can get up to summation. Now, tetany, this is if you're just holding your muscle flexed. Because in this case, the stimulation is, what we're saying is it's so frequent that the muscle just can never reset and it just holds eventually. This is like holding up something. And it seems easy at first, right? But then eventually you start shaking because your muscle is still producing this stimulate or being stimulated. It's still doing work this whole time. The reason it just stays up and goes up is that all the calcium that was in all of your muscle fibers, SR, it's out doing the work. It never went back into the sarcoplasmic reticulum. It's just out there. And this is kind of a cool thing, when you're doing stuff, that burning feeling is what you are feeling in that case, is that calcium is not resetting because you're stimulating things so quickly that you're not going to be able to, or it's basically not going to reset. Now, like I said, though, tetany summation, this is a good way to ramp up, muscles, and it's a very different way to train them too. So any exercise phys, that's kind of a cool way to kind of moderate things or kind of change things up. Okay, I know we're late in the video, but this may be the most important one because it's going to have a ton to do when we get into the cardiac muscle. And for whatever reason, people have a really tough time with this. So this is a good chance to like pause, take a break, get ready. Let's just do this quick, but make sure that you feel good about it. So in terms of actin myosin, it's a very simple relationship like we've seen. These giant myosin fibers in the middle basically have these little claws that are going to contract and bring in the actin, right? So we've seen that, like that big boom, that contraction like that. We see this all the way at the top right here. And this is just visualized. So all these little fibers are made of actin myosin. When they get told to contract, that's literally what they do. What was once, you know, maybe this length is going to turn into something like this length, right? That's the muscle contraction. That is an ugly myosin that I just drew, and that's not, sorry. Yeah, don't worry. Use the notes. But in any case, this relationship is best served by a certain physical distance, where right in the middle, you have the perfect amount of overlap, where actin is making really good contact points with enough myosin, but at the same time, you have all this room right here to still contract. Like your muscle can actually make that movement. So the amount of room it has to contract is going to result in a stronger, you know, whatever your action is. So let's look at when this is not optimal, because that's ultimately how I think it's easiest to teach this. When you have too much overlap of actin myosin, at first that sounds like a good thing, right? Like all the little myosin claws are going to be grabbing. We have a lot of overlap, that's great. But this is all the space that we have to move right here. So this is when you try and do some sort of movement, lift, anything, but you're in like a really bad position. You're basically like squatting too far down and now like there's no room basically. So if you put your muscles in a position where they're already like kind of contracted and pushed together, they can't contract any further. There's no room. Like this little contraction arrow right there, see how tiny that is? It's not going to do very much. So this is actually a really weak muscle position to be in. Similarly, now we're stretched too far. So all of a sudden, yeah, sure, we have all this room potentially that we could come down through and like it would be a big contraction. Problem is that the myosin heads have very little interaction right here with actin. Like they're barely grabbing it. See, all these contact points right here are really tiny. They're not, when you try to make this push right here, it's going to be really weak. This is like when you're trying to do some movement and you're already like fully stretched out, or you've pulled a muscle, for example. This is when things get really tough for you to like actually make that movement. Like if you're already fully extended on your leg and you try to jump again, it's not going to work. Your hamstring's already like fully straight at that point, right? When you jump, you like go into a squat optimal position and then boom, right? If you try, I'm not going to do it on video, but if you try to jump just flat-footed, just up like that, you can't do anything, right? Your muscles stretch too far. It's not in the position to actually bound. Similarly, it's really hard to jump when you squat all the way down. You've already contracted everything. You can't like, yeah, there's no room there. Okay. So that's how we can see this. This is going to apply a lot to the cardiac muscle. You'll see how. You can always read ahead. That's okay too. But in this case, what we're going to do with the frog is, so don't ask me questions on this because you better have watched this far in the video. Sorry, I know these are huge, but 2x me. Okay. What we're going to do is start taking like measurements of how strong the frog muscle is when there's no tension on it. So in this case over here, we'll basically increase the length of the muscle over time. But what's going to happen is that each time we measure the muscles, like basically tension, strength, when it's too short, like down here, it's not going to do much. So we're going to have the frog leg like on a pulley that's going to show us how strong is it being. It's not really going to do much at this really short length. But as we increase the length, because see how this is going to increase right here, you can start to see that as we increase the length, we start to get to this optimal stretch length, right? And so, you know, as we increase the strength, there's actually a position of the frog leg that's super really strong. You know, it's the perfect overlap of actin myosin. And we're still going to get a big distance of like smashing together on that contraction. Frog leg's going to do a great jump for us at that certain length. It was too short before. Unfortunately, as we keep stretching the muscle and keep getting data points, you don't have to get like 1,000 data points like this, but I am illustrating it. What you'll start to see is that as you overstretch the muscle, you lose what was good actin and myosin overlap. Sure, technically, if it could fire, it would be a big contraction because of all this space down here. But there's just like no interaction between actin and myosin. It's really tough. So as you ultimately stretch it, sad part of the lab, you actually will probably tear the muscle at this point because it's really far stretched, or pull it. And that's kind of, as far as a PT-OT type of thing, or an AT type of thing too, that's kind of like what a pulled muscle is going to look like right there. Just so far pulled that there's no interaction left. So kind of tough. All right. So at this point in the video, it's just going to be kind of some green text examples. You can stick around if you want, relax, whatever, or you can it and get ready for lab. So like I said, like nothing else is going to be on the exam. So it's kind of your call. These are fun examples, so I may as well run through them, but you're good if you're good on the video. Okay. So talking about muscles, everybody talks about slow and fast twitch. What does that actually mean? Typically, there's a couple genes that matter, but obviously environment's going to play a big role in the, what you were fed as a kid, how much exercise you got. But the basic idea is that with a slow twitch muscle, these are packed with mitochondria, energy burning, they're great. So that means that over a long distance, you can just keep going because your muscles are packed with mitochondria. They're going to keep feeding out ATP and you're just going to kind of keep going. That's great. The exchange that you make with fast twitch is that your muscle cells are hyper-packed with actin and myosin. What that means, and it's kind of an exchange, is that your muscles don't have a lot of mitochondria, unfortunately, so they burn out very quickly. But since they're so packed with actin and myosin, you have a lot more burst and a lot more like sprint type of speed. And there's a lot of us just in the middle that you're kind of okay. I did the 23andMe, for example, and I have one gene that says fast twitch. I have one gene that says slow twitch. So kind of makes sense. Unfortunately, I'm just kind of in the middle. So there's advantages and disadvantages to each. And it's kind of cool with Homo sapiens evolution that each had a big advantage in our biocultural evolution. Very cool. Again, green text because it's injury. Know the difference between ligaments and tendons. Typically, I would say like with ligaments, going from bone to bone, that's always typically a tougher tear, but I think maybe I'm underselling like Achilles tendon tears in that case. But as you can tell, tendons going to muscle to bone, ligaments are going to go bone to bone. So pretty nasty stuff. And in that case, and I mean, this is kind of a good detail. So with people getting faster, stronger, everything, why do we see ligaments and stuff tear? It's mainly because we can make a muscle bigger and like stronger, and it can like handle more stuff like that. You're kind of going to be born with this. There are things you can do to make it a little more elastic, a little better to stand stuff, obviously, but there's very few ways to make sure that this stuff is going to not pop, unfortunately. On that same note, this is basically the idea behind muscle recovery, is when you break things down, you need to repair that damage. That damage is typically repaired with amino acids, protein, but it's obviously a little more complex than that. This applies a lot to PT-OT, exercise phys. This is really good for healthcare and nursing too, just because in a lot of cases, patients, their main thing they need to do is just get out and get exercising a little more, even if that means walking, because this process right here is really important as far as the anti-aging process, because the body can tell if it's not being used anymore and certain systems start shutting down. That's kind of why you lead to a little bit of aging. Not being holistic or anything. That's literally what the processes are going into, but this is a blue text or green text slide, so it's not on the exam. So kind of last cool thing is that why is chronic exercise such a good thing? It's because when you repair the muscles and you go in here and say like, okay, like we need to repair, it's not just that you're going to build more muscles. It actually causes a very steady inflammation of your immune system. The immune system is going to come in during that repair process, because your immune system ultimately is a repair mechanism in a lot of cases. It's going to come in and kind of get stimulated and a test. And it keeps your immune system nice and like on point and says like, okay, like we're ready, we're healthy, we're good. We've been using ourselves like quite a bit as the immune system, so we should be okay. So a lot of this is going to tie into immunology and how cool that is. And that ties into microbiome and what, bacteria species live in your gut and all that cool stuff. It's all really fun. If you ever want to rent a book from me, let me know. All right. I feel pretty good. Let's go have a frog leg.